All right, people, let's get it. So in the last lecture, I discussed proteins and carbohydrates, and I talked about them in reference to their positions within the plasma membrane or on the plasma membrane. So in this one, I'm going to continue the discussion from the last lecture, but I'm going to add more details about the nature of those proteins that were mentioned. Also, I'm going to explain how these proteins aid in the process of diffusion. The way the membrane is structured does not allow for easy movement between the extracellular matrix and the cytoplasm. And this is very necessary, like it's so necessary. Homeostasis is what cells are constantly aiming for. If all of the solutes were able to freely move into the cell, and by solute, I mean like molecules that are not water. So we're talking sugar, salt, things like that. But basically, yeah, if all the solutes were able to freely move into the cell, there's a risk of organelles not properly functioning. When solutes enter a cell, they can cause major changes to the internal environment, so they can change the composition of the cytosol, and in turn, that could do things like throw off the pH. And one thing to know about proteins and how they work is that when you throw off the pH, they can denature and then they no longer work. So the enzymes in the cell working could potentially stop functioning, and that would mean trouble for a bunch of the metabolic processes that are going on. And so because the cell's not an open door, we call it a selectively permeable membrane. In other words, it discriminates between what should and what should not enter the cell. Molecules that have the same polarity as the inner part of the phospholipid bilayer, so the hydrophobic molecules, they can easily pass through the membrane and they can come into the cell. Things that are polar have a more difficult time crossing that barrier. So for example, here they talk about sugar. Sugar molecules, you know that they're carbohydrates, and one thing you know about carbohydrates is that they have a bunch of alcohol functional groups that are attached, and so they're attached to that carbon backbone along with some other hydrogens by themselves attached to the central carbons. And what we see is that they can dissociate fairly easily. That's why when we pour sugar in water and you mix it up, those sugar molecules kind of disappear. They've dissociated. So that dissociation can like carry a charge. If you knock some of the hydrogens off, you have these oxygen regions that are exposed that carry a negative charge, and so therefore it's polar. So we need sugar to carry out cellular respiration. That's one of the main ingredients. And if we need sugar to carry out cellular respiration, that means the cell needs that molecule. The regulation of the transport of things like sugar into the cytoplasm is still very necessary because there has to be a way to allow the entry of things like glucose. We don't want to completely keep it out, but we don't always want it rushing in either. So transport proteins are the answer to this conundrum. Basically, they allow molecules that have an affinity for water to cross the membrane. So there's a group of proteins called transport proteins, and they're integrated into the cell membrane, and they're capable of allowing things that are hydrophilic or water-loving molecules to enter the cell using the transport proteins as a conduit. And so one group of transport proteins that you have to remember are called aquaporins. So aqua like water and pore like an opening. So again, enzymes that have an opening for water to pass through. Those aquaporins, they're responsible for assisting the passage of water into the cell. Another group of transport proteins that are important are the carrier proteins. And these proteins are involved in the passing of molecules through the membrane. They get a substance on their surface and they change their shape, and the change that occurs is what moves the substance across the membrane. So we have carrier proteins that transport sodium and potassium ions across plasma membranes of nerve cells. And this is so crucial because without the carrier protein, you would not be able to have any nerve impulse conduction. and you need that in order for you to be able to react to things. The carrier proteins, they act selectively. They only are going to interact with a specific molecule or ion so that that molecule or ion can cross the membrane. So when we talk about sodium and potassium needing to cross the membrane and this carrier protein that's going to allow that to happen, we refer to that protein as a sodium-potassium pump. So the ability of a molecule to pass through a protein channel without needing any energy is called passive transport. And in order for you to get this concept fairly easily, it's easy to think of a person that has like a passive trait to them, or like they have a passive personality. So what happens with those kind of people is they don't get into confrontations, and they normally allow things to kind of slip past without making a big deal. And that's exactly how passive transport works. And it's happening because of diffusion. So in other words, the movement is happening because of diffusion. So diffusion is moving something toward a lower concentration gradient. So let's talk about or establish what a concentration gradient is. So if you were to look at a cell and you were to count the number of molecules that are on one side of the membrane versus the inside of the membrane, you would notice that maybe there are more molecules on one of the sides. So with diffusion, it's just a natural tendency for these molecules to want to kind of spread out from each other. And what happens is that causes something that we can predict, like a phenomena that we can predict. So the idea that you guys have already been introduced to is the fact that molecules move very randomly. But with this whole passive transport and diffusion thing, we're looking at the net movement of the molecules. So the net movement in this case would show that the molecules want to move from an area of a high concentration of said molecules to an area of lower concentration, and eventually the movement will kind of slow down, so a dynamic equilibrium is reached. And what a dynamic equilibrium is just it's the concentration of the molecules on one side of the membrane being roughly equal to that on the other side of the membrane. And once this happens, the net movement is zero or close to zero. And that's just kind of how that works. So here is an animation of diffusion.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
So there is the animation of that. And here in figure 5.9, we see what the diffusion of solutes across a membrane would look like. In this case, it's a synthetic membrane. When we do the national lab for osmosis and diffusion, and by national lab I mean the one that like all AP biology students are expected to complete, we use something called dialysis tubing, and that is a synthetic membrane. So you'll get to see this at work, not at the solute level, but definitely like taking the mass of the contents of the dialysis tubing being exposed to some water or I don't know, whatever other solvent mix we're going to place it in. You'll see that there is movement from an area of high concentration to an area of low concentration as far as the molecules are concerned. My puppies are killing me right now. They keep barking, but I can't stop anymore. All right, so in A, we're looking at the diffusion of just one solute. So there's water on one side of the membrane, and then we just have a bunch of solute molecules on the opposite side. So on the left of the first picture, that's where we see a whole bunch of that molecule, and then on the right it's just strictly water. So naturally, what the molecules want to do is to move away from each other. It's like being in a house party and there's too many people, and so you kind of want to just go outside to get some air. That's basically what the molecules are moving like. The second one, though, is showing you that movement starting to occur. So you have a couple of molecules that are now wanting to go into that open space where it was once just water. But you'll notice that there's this back and forth movement listed here, and that is that dynamic equilibrium that we're kind of getting at or trying to get to. At the very end, you notice that the arrow goes both ways, and it says equilibrium because the molecules are still moving across the membrane. But if we could take a snapshot, there would be roughly an equal amount of solute on both sides, in and out. The second one, though, is showing you diffusion between two different solutes. So sometimes we don't necessarily have just plain water to move into, and in a organism, it's not going to be pure water that, you know, is the space that is in the extracellular matrix. You always have some other solutes present. So the second one is a more realistic view of what would be happening. So in the first one, you notice that there are purple molecules on one side, so over here, and then you have these yellow molecules on the opposite side. They still want to move away from each other, and so they do the same kind of movement. The ones that are yellow want to move to the side where there are only purple ones, and the purple ones want to move to the side where there are only yellow ones. They want to get away from their partnered molecule, or like the identical molecule. So that's what's happening in the second one. You have net diffusion going both ways. Once we kind of get the purple ones on the left side equal to the purple ones on the right side, same thing for the yellow ones, that's when we have reached that point of dynamic equilibrium. And the movement is still occurring just as before. It just is that the net movement is very close to zero. This portion of the notes is confirming what you already have been told. So substances have a natural tendency to move away from the space where a bunch of the same molecule is being occupied. So in other words, if there's like a high concentration of a particular molecule in one area, the molecules naturally want to move away from each other or diffuse. So we call the action of like diffusion going down the concentration gradient. There's no energy required for it to happen. Like, this is just a phenomena that we have noted as we've observed solutes move. And then passive transport is what we have observed when solutes move across the cell membrane. Okay, so now that we've covered diffusion, let's talk about a specific type. So there's a name for the diffusion of water from an area that's saturated with water to an environment where there's less water, and we call that process osmosis, or that type of diffusion is called osmosis. Another way to look at the diffusion of water, or osmosis, is to describe the process as water movement from an area where there's less solute to an area where there is a higher concentration of solute. This animation is going to explain membrane selectivity.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
Osmosis animation.
The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis.
If you're looking at figure 5.10, which is the slide that I have up right now, what you see is a U-shaped tube that contains a selectively permeable membrane that's like directly in the center of the U-tube. So at first, the water level is the same on both sides. The only difference is the amount of solute on the right compared to the left. And when I say solute, I'm talking about the small green molecules that you see. They're labeled as sugar molecules. So that's what the solute is in this case. It's a carbohydrate. So over time, what we notice is that the number of molecules of solute does not change. They don't move through the membrane at all. What does change is the amount of water on both sides. So notice that the water moved to the side of the tube where there's more sugar, and that's what osmosis is all about. Here's a picture of it up close. You can pause here. You should count the number of molecules. Figure 5.10 is showing you close-up of the very bottom of the first slide that we were looking at with the U-shaped tube. So this is using reductionism to show you what things look like up close on a molecular level. You should pause and read the slide. So I'm going to end the lecture here, and I'll pick back up on the next one, going further into the territory of osmosis and what it would look like in living things.